A very good morning to all my dear faculty members. Welcome back. This is Dr. Balamurali, Organizing Secretary of this e-FDP and Assistant Professor, Department of Pharmacy Practice, MLRIP. A very good morning to one and all, and welcome to the fourth day of our 5-day faculty development program on artificial intelligence in public health, enhancing pharmacy curriculum and practice. After successful completion of 3 sessions, today we have Dr. Pankaj Kumar Singh from NIPER, Hyderabad with us. In the last 3 sessions, we have seen the impact of AI in community pharmacy and pharmacy practice area, different artificial intelligence tools available, and use of artificial intelligence in the medical field for diagnosis and treatment of various diseases. So today in fourth session, it is all about the targeted drug delivery to the cancer and role of artificial intelligence in it. I know everyone is waiting for today's session, so without any further delay, I would like to request Mrs. Shruti Reddy, Assistant Professor, Department of Pharmacology, to introduce today's guest speaker to our participants. Thank you.
Good morning, everyone. It is my distinct pleasure to welcome you all to this e-FDP session on targeted drug delivery to cancer, role of AI. Today, we have the honor of hosting Dr. Pankaj Kumar Singh, an esteemed expert in the field of pharmaceutics. Dr. Pankaj is currently an assistant professor at National Institute of Pharmaceutical Education and Research in Hyderabad, where he brings over a decade of experience in teaching and research. His impressive academic journey includes a PhD in pharmaceutics from CSIR, Central Drug Research Institute in Lucknow, and a postdoctoral fellowship at the Institute of Nanoscience and Technology in Mohali. His expertise in targeted drug delivery systems, particularly for cancer treatment, is complemented by his practical experience in quality assurance with outcome drugs and pharmaceuticals. Dr. Pankaj has been involved in numerous impactful projects funded by prestigious organizations, demonstrating his commitment to advancing pharmaceutical sciences. With over 85 publications in high-impact journals, 7 patents, and active participation in various research initiatives, Dr. Pankaj's contributions to the field are truly commendable. His research focuses on innovative drug delivery systems, including peptide-based liposomes and ligand-anchored systems, which hold great promise for treating life-threatening diseases. Today, we are excited to learn from Dr. Pankaj as he explores the intersection of targeted drug delivery and artificial intelligence. His insights will undoubtedly enhance our understanding of how AI can revolutionize cancer treatment. Please join me in extending a warm welcome to Dr. Pankaj Kumar Singh. We are eager to hear your thoughts and insights on this important topic, sir. Thank you.
I have so this vision and mission of NIPER Hyderabad. So vision of NIPER Hyderabad to serve as a leading global institute in the field of higher education and research, in pharmaceutical sciences, management, and the medical devices. And we are going ahead with this vision and with the mission to strive toward the excellence in the field of education, research, in basic sciences, management. This is our vision and mission, and we have a very good opportunity to be located in the NIPER in the Hyderabad. That is one of the hub of pharma. So that is a great thing for NIPER Hyderabad, which makes difference. This is the journey of NIPER Hyderabad. We started with the 2007, with the MS in 3 streams: medicinal chemistry, analysis, pharmacology, and toxicology. After that, in 2010, we introduced pharmaceutics, as well as in 2021, we started PhD in medicinal chemistry, pharmaceutical analysis, pharmacology, and toxicology with Sutix. And another big thing came in the picture when MBA started in the 2012. 2013, we started MTech in the RT and PC. In very recent 2020, we started MTech in medical devices. 2021, we started National Product, Pharmacoinformatics, and Regulatory Affairs. So these are the total journey of NIPER Hyderabad in last approximately 15 years. And this is the one of the our achievement in last 7 to 8 years. We started with the 5th rank in the 2017, and in last 3 years, 22, 23, and 24, we are managing a good rank in the NIRF ranking, which is one of the prestigious ranking in the our country. And the best thing is that when I am presenting, I am the nodal officer of this NIRF ranking for Naipur Hyderabad. So always when I'm discussing all this, my area of work is nanoparticulate system, nanomedicines. So one of the classic picture which is showing the different type of nanomedicines, which is 2018. Grateful for this image, and it's very inspired me. There are different type of nanomedicines. Conventional, it may be used as a theranostic, means we can explore this nanomedicine for different type of function, or different type of use, different type of application. We can so in that basis we can categorize our nanocarriers in the 4 different part. The one is conventional, second is theranostic, ligand targeted nanomedicine, and pegylated nanomedicine. So in conventional, if you use some triggered release coating, like if you use a pH sensitive polymer for the surface of that nanocarrier, it will be becomes as a targeted moiety, targeted nanocarrier, which is able to release the drug, or which is able to destroy in the environment which is favorable for that pH sensitive polymer. Is it? So this approach is well explored, and people are still exploring this approach, in which they are using some pH sensitive lipids for the development of liposomal formulation, and they are targeting to the specifically to the cancer by a strategy that cancer cells having quite lower pH than in normal cells. Quite acidic environment is there, so that environment can be targeted, or can be so basic pH that driven by increasing the temperature of that particular organ. For example, a person, if you're having a solid tumor in breast or any other part of the body, so we can locally inject this type of formulation, which is having the thermosensitive type of lipid, and by using some temperature from the outer side of the body, we can make sure that that formulation is start release of the drug from that carrier system. Similarly, we can also explore this nano carrier by using as a theranostic to get for the diagnostic therapeutics, by using some targeting ligands, by using some functionalized imaging agent. That imaging agent can be used as a diagnostic agent for that particular cancer. Similarly, if we use some ligands, like if we use some peptides with antibody, which is specific toward that particular cancer. For example, a person which is having the brain cancer, and this brain cancer or breast cancer or cervical cancer is overexpressed with the different receptor. So we can explore the different ligands, which is quite particular about that receptor. This thing will targeted your nanomedicine or nanoparticle toward that particular organ, particular cancer. So that is going to be that is already explored a lot, and people are still exploring the use of different ligand, just such as carbohydrate for small molecules. They are using antibodies. They are using protein, and the selection of these targeted ligands are quite critical in this type of nanomedicine. On the basis of selection, we can make our formulation to targeted toward that particular organ or particular cancer. We can also explore pegylated type of nanomedicine to increase the circulation time in the body, and already few formulation are in the market which is pegylated. These are the some of the market formulation. These are this formulation can be a motivation for us to do something in the area of cancer nanomedicine. You can see, I think all of you are aware about the doxil, which is a formulation of doxorubicin, which is in the market, which is, I think you are also aware about the ambrexane, which is a formulation of paclitaxel, which is already in the market. So this type of formulation, or we can say these are the type of product which in the market can be a very good motivation to do something in the area of cancer research or area of targeting cancer. So these are the different market formulation. Is there on video there, that is a formulation of inoticon, which is a liposomal formulation in the market. These are this is a list of different pharmaceuticals marketed nanoparticle system is available for the cancer research in the market, and basically these all are lipid based nanoparticles. So as well as we are also exploring in our labs, we are exploring the lipid based nanoparticle. So this is a total type. These are the different type of nano carriers or market formulation which can be explored more, because all these formulations are not targeted toward the that particular cancer. That is one of the loopholes which is associated with this type of formulation. Is it? For example, if you see about doxils, it is pegylated liposomal formulation, but it is not targeted toward that particular cancer. Is it? It's increased the circulation time of in the body, but there is no targeting ability in this formulation. So this can be a area of research. This can be an area of work which we can do. These are the different methods. If we take one by one, liposomal formulation, we are exploring a lot. And this we are using different diagnostic agent in the liposome. In the most left side, you can see there is a conventional liposome. By using some diagnostic agent, hydrophilic agent, hydrophobic molecule, because by using some antibody, we can make this liposome more specific toward toward the cancer cell. Is it? Not toward the normal cells, and ultimately it will be reduce the toxicity related to all this formulation. All this formulation, what we are able to see in the right side of the picture, doxils is there, ambisome is one of the another liposomal formulation which hold a good market for the amphotericin B. It is used for the used as a antifungal treatment, as well as treatment of viscerous masses. So this type of formulation can be a mark bench for us, and we can do more in that area of work. We can improve the targetability of ambisome. We can improve the targetability of doxils. So these are the area we can do, and we are working. So these are the different method we can say. You can see there is a method of thin film hydration method. One is small injection method. We can also develop by using some freeze drying method. So these are different method how we can develop the liposomal formulation. and these all are possible at the at laboratory. So we can explore this time, and we are exploring in our labs all this formulation. Similarly, if you tell about the polymeric nanoparticle, as we discussed about liposome, polymeric nanoparticle, also there are a lot of methods to develop these polymeric nanoparticles. Out of that, one nanoprecipitation is one of the method. By in that, we have to solubilize our solvent polymers, drug plus oil, what in some organic phase, and we should have a aqueous phase which is having the water plus surfactant. Basically, surfactant is required to stabilize the complete formulation. So these are the different method. There is emulsion co-resorption method is there, which we can explore. Double emulsification method that we can explore. So these are different method by which we can develop the polymeric nanoparticles. And the polymeric nanoparticle based formulation is in the market, which is Ambrexel, which is right side of the slides. Another nano carrier is micellar formulation. This is one of the nano carriers which can improve the which can easily improve the solubility and viability of various hydrophobic drugs. That is which is the one of the uh, uh, one of the problem which is associated with the hydrophobic drugs is solubility as well as viability. So there are different methods for development of micellar formulation. We can use this diblocopolymer. We can add in the water, and that is the first method, direct dissolution method. In that we can use diblocopolymers. If you add in the water system, it will make a self assembly. Otherwise, you can also go with the dialysis method. In that, polymer and drug can be dissolved in a organic solvent, and which can go for the dialysis against the aqueous phase, we can say, or water phase. So you can get micellar formulation inside the that dialysis bag after the movement of water phase in the toward the dialysis bag. Similarly, other methods are there. Solvent evaporation methods are there. Oil and water emulsification method there. If you use this polymer, if you dissolve this polymer or dibloco polymer with the drug in a organic solvent, and if you evaporate this in a rotary evaporator, you will get a thin film which can be reconstituted with some water phase, and you will get the micellar formulation. So I just want to give a brief about the different nano carriers, what we are exploring, and what other scientists who is working in this area can be they are exploring to develop the nano carriers. So this is the current innovation in the area of nanoparticle system. Again, I am saying we can explore synthetic type of polymers. We can explore, and people are also working on the gene therapy. People are also working on the immunotherapy by using the nano carriers, by using different liposome formation, as well as they are also working on the cancer therapy. So these are different area, as well as current innovation, which is related to the nanoparticle system, and we can also use. So you can see the challenges with the nanoparticles. Still, it's having a lot of advantages, but there are still some challenges are associated with the nanoparticle system. So its route of administration, biodistribution, biological barrier, toxicity. Still, it's having some toxicity issues. If you see about the metallic nanoparticle like gold nanoparticle or silver nanoparticles, this type of nanoparticles are highly associated with the toxicity, which we can say it is going to be in the body for a longer period of time, and start giving some toxicity to the body. So these are challenges are associated with the nanoparticle system. And now we will discuss about some intelligence, is it? And so artificial intelligence is going to mimic the human intelligence and human cognitive function like problem solving, learning, and it's basically it's artificial intelligence is used to predict and automation. If you see example, if you are having for as a chemist or as a medicinal chemist person, if you have 100 number of synthesized molecule in your library, or 1,000 number of synthesized molecule in your library, which is having a particular structure and particular properties. So it's quite no, quite impossible, we can say, to check all this synthesized molecule, all these 1,000 molecules on some cell-based assay or some animals. It is quite impossible to check the activity of all these things. But with the help of AI or with the help of some machine learning tools, we can put the data related to that 1,000 or 100 of molecule in that particular AI or AI model or machine learning model, and we can put the data rate. For example, we can put the structure, we can put their solubility profile, we can put their the number of double bond present in that particular molecule which you synthesize. So this different data we can put in our library, and that AI tool will predict on the basis of data which is already in the wave. It will make itself trained to find out for the particular disease or for the particular cell line or for the particular therapy, which type of molecules are already got approved, or which type of what are the different properties a molecule should have to give the a particular type of efficacy. So on that basis, definitely this AI tool will give you a good number of results or good number of predicted molecules or of molecules. It will give you 5 molecules on ranking basis. You can try these 5 to for the treatment of cancer or for the treatment of in vitro efficacy, for checking in vitro efficacy against some cancer cell line. So that these things are going to be improved, or these things are going to be very useful. We are working in the nanotechnology, as well as while we are working with the help of while we are working for any treatment or selection of any drug, or selection of some lead from number of molecules. All of you are very much aware. Means this is a quite old thing. When if you are starting with a 5 molecule from the pre-formation and completion up to, at least I am saying, at least up to the in vivo efficacy assessment in a animal model, it's quite costly journey from starting from the pre-formation and complete up to the even up to data. So AI with ML tools are going to be very helpful. It's increase the efficiency and reduce the cost of the drug research. For example, after the getting that 5 molecules out of 1,000 or 100 molecules, we can again go for the in silico toxic studies or in silico tools we can use, which can give the most promised or most promised drug or most promised molecule out of that 5, which can be which can a very good interaction between that protein and that particular synthesized molecule. So ultimately, these are going to be increase the efficiency of your work and reduce the cost of total your nanoparticles research. So these are the different application. AI can help in the advancement of nanomedicine. AI is very helpful to find out a particular polymer, which is very much useful. AI is very helpful to predict the predetermined goals. Okay, if you are going to work on a nano carrier, and if you want to work on a brain cancer, and you want to finalize your predetermined goals before going to start your research or before going to start your project. You can take the help of different AI tools. It will search from the database in the last 20 years. These are the different research happened, and they are concluded that a drug or the formation which is having the size between the 180 nanometer to 100 nanometer will be the most suitable particle size to get that particles or get that nanomedicine distributed in the brain. Is it? So that will be your predetermined goal, to get a particle size of 150 nanometer or 100 nanometer, and particle size between the 150 to 180 nanometer. Again, the same AI tools will also help people are explored. These are the different nano carriers. They explode liposomal formation, they explode nanoparticle system, they muscular formation. But the maximum number of research which is in the web or in the scientific web is the development of liposome with the particle size of 150 nanometer, with the PDI with the 0.1 or 0.2 PDI is the best suited tool to deliver a anti-cancer drug for the for the brain or for the brain tumor. So this way we can get a lot of help in the AI tools. Okay, we can also predict the release kinetics of our formation. We can also estimate the permission and cellular response of our drug. We can predict, we can see, we can find out the shape as well as size of our nano carrier what we already developed. If you put that images which is having a 10 number of particles or 100 number of particles in their that picture, it will give you the distribution of particles, how your particles are distributed. It's having it's very dispersed in the size, or it's very narrow in the size, and how much it is dispersed. That thing you can find with the help of we can find out with the help of, we can predict with the help of AI. Even there are some AI tools are already available, which can predict the toxicity of your developed formulation or developed molecules. So and all these are based upon its scientific data, or we can say scientific pull data in the scientific hub or scientific web. So these are different applications which we can find out with the help of, which we can use while development of nanomedicines. So basically, this is the state of art in the AI assisted nanomedicine. It's going to be mine the data from the web or scientific web. It's going to collect a huge number of data. It's going to screen this data in such a way so it can tell you, or it can predict something. So there is a many lakh type number of papers are there, research papers they are there, which is basically based upon the different nano carriers. And so it will make this all data in a proper manner, so it can find out if a person is going to develop a liposome with phosphatidylcholine plus cholesterol up to a size of 200 nanometer, and if it is going to use polysaccharide as a ligand over that, so what will happen? So it will make the data in a such a way so it can conclude, and it will give you a result on that. So you have to just give a write down in that system or with that AI tools. I need to develop a formulation with 150 nanometer with narrow PDI. Narrow PDI means 0.1 to 0.1 or 0.2, and it should have a negative charge over the particles, and it is good in the uptake of some cancer cell line, MDMA cell line, or any MCTs or HCH, these different cell line, it should target it toward that particular cell line. So if you add this data, so it is going to find out this data, or it is going to screen its data which is already present in the scientific web. From there, it will give you a result that you can use phosphatidylcholine with cholesterol with the ratio of 8:1 by using acetone as a solvent and water as a aqueous phase. The ratio should be 10:1 with the steering speed of 800. So this is the different parameter you will get from this AI tool. Now this is your work to, or your part to do that experiment and check the suitability of that particular AI prediction. If it is very close, means you are it's very the prediction of that AI tool is very much accurate. Okay, so it will be very useful tool if you are going to start with work with the 5 different polymer or five different lipid with the 5 different drug with the different parameter. It is a very long process to get a optimized formulation. But if you use this type of tools, this is going to be reduce your 90% of your work, because getting a uniform particle size and predetermined nanocarrier is one of the challenge while development of nanomedicine, or while development or while working on the this different nanoparticle system. So these are different optimization, how we can optimize the nanomedicines. So this is a totally this artificial intelligence is a approach which is having different part. It's having the robotics, it's having the automated planning, it's having the one part is natural language processing, it's having multi-agent system. So these are different part of your artificial intelligence approach. And these are some very classic examples of research which they use for the different AI method for their nanocarrier development and for their formulation development. So one is that the first one is they develop the nanoparticles, and they use the machine learning as a tool. And they actually, by using this machine learning tool, they find out, they forecasted their mean particle size as well as PDI, and they also compare the various factors while development of nanocrystals. This work got published in 2020. In that, they find out the different forecast their second one is of nanofiber and recognize the production and progressive flaws. So while developing nanofiber, use this machine, they can find out what is the parameter we are not getting, or what are the parameter that is going to affect the outer surface of the nanofiber. And third one is they are working on the solid lipid nanoparticles. They use this interaction potential of CPP into 3 different alternative phospholipid monolipids while development of the nanoparticles. So there are different methods they already people are already explored. Out of that, I will discuss one of that. In that, use machine learning for the optimizing nanocrystals. So they use 2 approaches: top-down approaches and bottom-up approaches. In top-down approach, they used ball milling, high-pressure homogenizer, and anti-solvent precipitation method. These are the different methods which we can we can use for the development of nanocrystals. In that, they collect a huge number of data from the scientific arena, which is used for the development of nanocrystal. From that, they find out there are 910 nanocrystals size data is available with them, and 341 PDI data is available with their three different preparation methods. So these are the three different methods which they are going to explore, and their goal is to find out the best suitable method by which they can develop their nanocrystals. So the first thing is to get the useful information or a huge data related to the nanocrystal which is already available. So they got they collected this from Web of Science and their in-house data. Okay, but this all nanocrystals are related with the ball milling, high pressure homogenizer, and anti-solvent precipitation. So out of these three different methods, or all this data what they are collected, they use this data to 80% for training subset. Basically, they want to stabilize some more for their research. They use 10% of their data to validate that model, and they use 10% of their data to test their model or test their work. So they developed this 8 different learning model, machine learning model, that is based upon the Python, and they find out ultimately they are getting the lowest particle size from the light gradient boosting machine. That is one of their predetermined work. They want to be a smaller particle size, we can say, and the error is minimum with the first model, which is this is fourth model, which is light gradient boosting machine, and further they utilize this machine learning tool for further processing. And by using this different by using light gradient boosting machine, they find out these are the different parameter which is going to be impact vigorously to the development of nanocrystal. So the first parameter or most prominent parameter which they got is milling time. So milling time is one of the parameter which is going to affect the nanocrystal size most, as well as their stabilizer, what stabilizer they use, their molecular weight of their API. So these are the different parameter which is going to affect the development of nanocarriers most. The most prominent is milling time, with the second one is stabilizer 1, molecular weight of API, stabilizer 2, rotation speed. So this is the ranking of the effect which is affecting a lot while development of this nanocrystal formulation. When they are developing the same formulation with the high pressure homogenizer, the cycle index is one of the most prominent factor which is affecting concentration of stabilizer 1, API. These are the different factors with the ranking is affecting a lot while development of nanocrystals. While anti-solvent precipitation method, again, the stabilizer 1 is one of the most prominent factor which is affecting the development of nanocrystal, while the concentration of API, melting point, these are the other factors which are also affecting the development of nanocrystals. And when they do all this experiment, okay, suggested experiment from these different tools, they developed celestial nanocrystal formulation. They prepared by all these three methods: ball well milling, high pressure homogenizer, and anti-solvent precipitation. And they find out the predicted value. They actually compared the predicted value with the experimental value in the term of size, by all these three methods. If you are able to see, all this experimental size is in the right side and predicted value is in the green color. So if you see, there is a quite similarity, we can say, or predicted value and experimental value quite similar in the term of, when we are saying about the ball weight milling and high pressure homogenization. While in the anti-solvent precipitation method, there is a huge difference. Even it is two times difference in the experimental value and predicted value from this AI tool. So, and they also mentioned the problem, or we can say the result what they are getting with the anti-solvent precipitation is due to the having a very less number of data available for anti-solvent method, and there is insufficient satisfactory prediction is attributed with the insufficient data and volume and size of the PDF, because the maximum uh, number of articles or research which is available in the this scientific arena is with the ball valve milling and high pressure homogenization. So due to the less number of data available in the web, it's there is no there is a huge changes between the predicted value and experimental value of the developed formulation. Again, they do the study of their developed formulation. We can say they also compare with the pure drug. Okay, pure drug cholesterol with the second method, first method ball milling, second method high pressure homogenizer, and third method anti-solvent precipitation. So you can see there is the nanocrystal which is developed by the anti-solvent precipitation method are showing the amorphous form in XRD, while other two methods, ball milling and high pressure homogenizer, is showing some crystalline behavior in that when we are seeing the XRD results. While in vitro dissolution, there is quite similar pattern in all the developed formulation, and all are showing the quite similar type of dissolution pattern with respect to the pure drug. Okay, so ultimately, just I want to say that this one of the this classic example or best example of using of AI and machine learning, and you can see this all these tools are reducing number of experiment. If a person will start working with the all these three method with all this work, so it will require a longer period of time. While if you see this paper, you can in future, if you want to work on the nanocrystal development or any other type of research which is related nanocrystal, it will require very less time. You can find out, you can go with you can go with the these two method, ball weight milling or high pressure homogenizer, or you can choose this milling time as your prominent factor. You will in your optimization, you will focus on the milling time, or you can focus on the stabilizer which you are using. You can also focus on the API. Okay, so it is very useful to do the further research if you have this type of data, if you have this type of tools, AI tools or machine learning tools. For can I ask how much time I have more, because I have many other research project which is on my lab. Can I? Hello.
Yes, sir.
Can I have 10 minutes more?
Yes, sir.
Okay, fine. So I will also discuss about some research project which is currently working in my lab. So one of the most prominent work in what we did in the last 1 or 2 years on that is based upon the rheumatic arthritis. That is we used in that halozone as a gel and tofacitinib citrate as a as a functional excipient. This is the mechanism of action of tofasolid, that is a JAK inhibitor. Okay, and these are the right side. We can see there is some market formulation which is already in the market, which is related to tofasolid. There is an extended tablet of 100 mg. One is 5 mg tablets is there. 1 mg tablet is there. 1 mg oral solution is there. So these are the different formulations. In that, we choose hyaluronic. Basically, in hyaluronic, we are using hyaluronic acid in the core of liposome, and we gave this carrier system as a hyaluronic, and we use tofasolid citrate as a API, and ultimately this liposomal formulation, which is having hyaluronic in the core, we added in the one of the gel, that is we optimize. So this is the formulation development. We use solvent evaporation method to get the and the particle size zone is nearby 85 nanometer, and particle size of tofasolid loaded is 181 nanometer, with the entrapment efficacy of 59%. This we just checked with the skin permeation. If you see the skin flux of developed formulation and plain gel is very much different. Tofasolid loaded halogen 3 times better than plain gel. And this the pH of our formulation and viscosity and visual appearance of our formulation, and most from of this is in vivo efficacy, in that we did and we divided this all animal model in different groups, and this is the prominent result of this developed formulation. See the difference between the power of formulation and other group. The normal control is having the very, while this formulation is the having maximum power volume due to the inflammation and due to this rheumatic arthritis. While if your developed formulation is having a medium, or this blue one is tofasolid boric acid hyaluronic gel, which is having the most nearer, we can say, or most less power volume compared to the disease control. This is the pictorial representation of the developed formulation. You can see this. This is the normal. This group is normal control for different days. This is the disease control. You can see the power and the use in the that particular group, particular animal model. If you compare this with our developed formulation, which is this one, you can see quite similar to the normal control after 28 days. Initially, it is also having very inflamed, but with the treatment up to 24 days, it's got treated with the as compared to the disease control. Okay, so this is the one of the most prominent result of what we get this study. This is the X-ray analysis. You can see the normal control and disease control. There is a huge difference in the there is a banding of this area. While if you see our developed formulation having quite similar, not I can say it's 100% got treated. It's quite similar to the so we want to see that animal model, how walk there after the diseased or after a normal with the control. So they are also very much similar of our developed formulation with this normal control, and other you can see there so much variability in the walking track analysis. studies. This is a strategy we perform. This is we perform with the help of some of our biological sciences peoples, and we can see there is a increased edema in inflammation of immune cells, which is indicated with the these marks. So this is the one of work we got a lot of hype. We got our patent got published, and this is the one of my student who did this work, and she got many awards for this. And we are still hope with the hope, and we are still discussing with few industry for this work to get commercialized. And this is another work, so a lot of things are there. So I think it's enough for today, and thank you so much for inviting me. And there is a lot of research there which we are doing in our labs. If anyone want to really discuss with me about all this work, you can mail me or you can call me anytime, so I will be available to discuss all these things. And at last, I just want to thank you to all of you. And this is my research group and PhD scholars who is working with that. I acknowledge them because they are the pillar of lab, and they are working very hard to get all this research on everything. Thank you so much.
Yes, sir. Thank you very much for your informative session, sir. And number of case studies, what you have explained, I hope it will be how it explains how effectively it helps in reducing the cost and as well as time in the cancer therapy. Sir, I have one query, sir. Here, how that AI will assist in monitoring cancer progression and patient response in the treatment, sir. I hope you have explained with respect to the formulations, many case studies you have explained.
Mm-hmm.
And further, once the drug has given, that nanoparticle liposomes are given, how that AI will assist in monitoring that cancer progression and as well as the patient response in the treatments.
Even nowadays, you are the cancer response. Nowadays, there is a diagnostic tools are there which is totally based upon AI, and on the basis of pictures, on the basis of X-ray image of that particular person or particular patient, that AI can detect that person is really affected with some breast cancer or brain cancer or what. So that picture even there, and that is based upon the pictures or topography or the surface morphology which is already available in the way. So by using that information, they prepare a tool, and by using that tool, they can predict the size, they predict the severity of that particular cancer. And someone is asking for my email ID, so can I write in the chat? In chat, I put my email ID, that is a personal email ID. So anyone can make for the query paper. Yeah.
Am I audible to you?
Yes.
So I need to know as there is the workshop on AI in the detection of, you are mentioning that it is being used in the detection. So could you please tell me what web server is there which we can use for the detection of cancer, or what software can be used in the detection of cancer? Can you please highlight this thing?
If you just do one thing, mail me on this mail ID, I will send the complete data, complete information about that tool which is used for the detection of cancer on the basis of some AI and based upon the AI ML background or AI ML as a tool.
Okay, sir. Thank you.
Okay, thank you.
Dear participants, if you have any queries, you can ask our speaker. Yes, sir. There is a question from the participants. Can we formulate cosmetics with nanotechnology?
What project I just discussed with this last rheumatic arthritis, that is one of type of gel, is it? That is going to be applied over the skin only. So it's very much possible to prepare the gel system. So it's very straightforward. If you are going to start a work on the gel or any solid system or solid system, definitely you have to think about a lot of number of polymers, which what will be the base of my gel, what will the concentration of gel. This tools will give you exact figure if you use this particular polymer with this concentration, get this much of viscosity, and you can use for the disease, or it will give this much of permeation. So it will help you to get that one. Now, when you have this data, you can develop that formulation in your lab, and you can how much is correct or how much it is similar to the your given data. What is the predicted data and what is your experimental data you are getting. So at least you will be at 90% of your optimization when you have this data.
Okay, sir. Thank you for that. There is one more question. How DL model is helping in targeted drug delivery?
Hello?
Hello. Yes, sir.
Uh-huh.
Neeraj, sir, there is one more question from the participants. Neeraj,
Hello?
sir, am I audible?
Yes. Yes, you are audible.
Okay, sir. So the question is, how DL model is helping in targeted drug delivery?
What does mean by DL model?
They have mentioned how DL model neural network is helping in targeted drug delivery. This is the question they have put in the chat box.
No, that's what I'm saying. This all this even a nano carrier or nanoparticle system. For example, as he's saying, the targeting of cancer. So targeting of cancer can be do by the two ways. One is by passive targeting, or one is by active targeting. So if you find out the wave of sign of wave, you will get that if you particles are having the size below 200 nanometer, they are able to show, or they are able to give the EPR effect. So in that aspect, and again your question will be, or your expectation will be, what are the different polymer by which I can develop a nano carrier which can give the particle size of 200 nanometer or below 200 nanometer. So that will be your another query. Question will be, what are the different ligand which we can explore to target the breast cancer or target the brain cancer. So this type of things you will get from the AI, and you can start from that point. For example, that AI will give you can use PLGA to get a nano carrier or to encapsulate a cytotoxic drug. with the size of 180 nanometer. You can use chondroitin sulfate, you can use hyaluronic acid, you can use phospholipid serine as a targeting ligand to target the CD44 receptor or any other receptor. So this type of things you will get directly, and now you can explore the given data, okay, or predicted data, and even this is less. I'm saying you will get more than that. It will also give the concentration of the polymer. It will give the solvent which you can use. You can also get the what are the different stabilizer you can use. So this type of data you can use, and you can develop your formulation to go get your predetermined goals.
Okay, sir. Thank you.
Sir, actually, you have explained many case studies. Out of that, while applying this AI in the cancer therapy, what are the limitations and challenges we are facing, sir?
Limitation means uh, we we will get the predicted value from AI, but at last we have to do experiment, and we have to see the suitability of that given a predicted value. For example, if you when the things will come with the biological system, or when it will come to the interaction of a formulation with a membrane of nucleus membrane or cell membrane. So that type of data we have to do. Again, we have to perform the experiment, and we have to come with the conclusion. Again, when I discuss with one conclusion or one case study, there is a very less, or we can say there is a very less similarity in the predicted value and experimental value. Okay, so there is a huge change in that. So this type of things can come into the picture, and you can not get the right values or right predicted value. Is it? And it's depend upon the data availability and how much data available in that for that particular experiment. And like nanocrystal I discussed now, ball milling method and HPH method is so much explored. So that data is very much similar of predicted and experimental. While the third method is data is very less. That's why there is a less suitability or less similarity in the predicted and as well as experimental value. So this type of challenge definitely come, but it will definitely reduce your time and cost of your research.
Sir, can you expect it as helps that reducing time and cost of that when you are applying that AI in the cancer therapy, sir?
Can you repeat the question?
Can you expect how effectively that AI will be helps in reducing cost and time in the cancer treatment?
I can give you a
The
simple
cancer
example.
therapy or drug discovery,
I can give
clinical
you.
trials, whatever you are applying, how
Mhm.
that AI will helps in reducing cost and time, sir? How far?
That's what I'm saying. I'm just giving you a simple example what we did during the corona time 2023, 2022, and 21. We have a student with us, but we don't have supply of chemicals. All students are. We are thinking to what to do. So at that time, planning to explore the eclabrutinib as a drug that is approved from other disease, approved for different cancer. So at that time, we use this AI tool, this molecular modeling tool, and we explored this eclabrutinib for different type of protein or different type of cancer. Protein which is related to cancer, and we find out that it can be also useful for the treatment of lung cancer. So this type of work we can do, and this type definitely now a person can see this paper or see this result, and he or she can explore the activity of eclabrutinib with the brain sorry with the lung cancer. So how in this manner definitely it can be helpful. Even also improve the quality of research, or we can also improve we can give a new area of research by using this. Even you get any student who is not having the access of all chemicals, different instruments, they can think, they can see how a anti-cancer drug, which is approved in 2024 or 23, by using of some molecular docking tools, and they can explore the interaction of this new drug, and with the different protein, with different cancer associated protein, and they can find out, oh, this drug can be also useful in this different type of cancer or different cancer.
Yes, sir. Thank you, sir.
Good afternoon, sir. So in case of cancer diagnosis, we use different images by using convolutional neural networking for the prediction of the stage of cancer. What should be the optimum magnification which is required for image analysis?
Actually, I'm not this much expert in this in image analysis and all these things, so I am not the right person to answer this. I'm not that much deep because I'm expert in this how to optimize the formulation, how to get the particular particle size, but image analysis I'm not aware.
Thank you, sir. Participants, are there any other questions? Otherwise, we will wind up the session for today with a vote of thanks. Okay, sir, there is one more question. Can we do nanotechnology in the ordinary lab? What equipment is required?
Yes, you can send, if anyone having this type of queries, they can come to us, and they can visit once our lab. These are the different means equipment are available only, and no labs is having all the equipments for everything. In that case, please collaborate with different institutes, you know. Even rotavaporator is required, sometime is required, sometime simple steerers are required. So it's required, means it's not like that in your lab everything will be you will have everything, then you can start working on the nanotechnology. You have to collaborate to everyone. Okay, thank you.
Thank you, sir. Okay, now I request Mrs. Pravalika ma'am, Assistant Professor, Department of Pharmaceutical Analysis, to deliver a vote of thanks.
Thank you, Balu sir. As we conclude our enlightening session on targeted drug delivery to cancer, role of AI, I would like to extend a heartfelt gratitude to Dr. Pankaj Kumar Singh for sharing his invaluable insights with us today. Dr. Pankaj, your expertise in targeted drug delivery and the integration of artificial intelligence has provided us with a deeper understanding of these critical topics. Your extensive research and practical experience have greatly enriched our knowledge and inspired us to explore innovative solutions in the field. I appreciate the time and effort you took to engage with us, answering our questions and sparking meaningful discussions. Your passion for advancing pharmaceutical sciences is truly inspiring. Thank you once again for your contribution to our session. We hope to have you with us again in the future. I also thank our dear management, principal, vice principal, and the organizing secretaries for giving us the opportunity to be part of such an informative and enlightening session. Thank you all for your participation.
Thank you, Pravalika ma'am. Okay. Thank you, Pankaj sir, for attending our FDP program and talking about the AI, very specifically how we can utilize that for cancer treatment. Thank you so much for that, sir. And also, I thank all the participants who have attended for today's session. So with this, we are going to stop today's session and we'll meet tomorrow with the last session of our 5-day faculty development program. Thank you. Thank you very much, all the participants. So you're receiving the feedback link to your mail IDs as well as in the WhatsApp group. Please share your valuable feedback. Thank you once again.
Thank you, everyone. Thank you so much.
Good, sir. Good, sir.
Yes, sir.
Pankaj, thank you, sir. Thank you so much, sir.
Thank you, ma'am. Thank you,
Yes,
sir.
sir. Thank you, sir.
Can I leave now?
Yeah.